Hello everyone. After talking about different properties of materials, we have briefly talked about bulk properties. Now we will talk about surface properties of materials and how these surface properties are characterized using different techniques. Talking about surface properties, actually the material surface is the one that has the very close contact or intimate contact with the physiological system. The atoms that make up the outermost surface of the biomaterial drive many biological reactions that occur in response to the biomaterial. For example, on the surfaces, there occurs protein absorption occurs, cell addition occurs, cell growth also occurs, and if the blood comes in contact with the surfaces, the surface should also be the blood compatible. As I already said that surface are in close contact with the physiological system, so it is important to study the biocompatibility of different surfaces or different materials. So to study the biocompatibility, we should also know the influence or what is the reaction of these surfaces, or what are the influence of these surfaces to different proteins, different cells, and different organs of the body. And when this biomaterial surface firstly comes in contact with the body, the cells, they first interact with this surface and responds to the surface. That is why we need to understand the surface structure of the biomaterial. The surface of the material is also dynamic in nature. The surface properties or the unique reactivity of the surface is it may change after some time, maybe due to the surface oxidation or different chemical reactions that occur on the surface in different conditions. For example, when it is outside the body, there may be different conditions when it is inside the body in a physiological condition. Because of the different because of the different interactions, the surface properties may change because of different chemical interactions, so that also needed to be considered, and those interactions should also, and those interactions and those reactions should also be studied in the physiological system. As the surface are in close contact with the environment, so there are the chances of the contamination. So surface of the materials are usually contaminated, or they are readily contaminated by the components from the vapor phase. For example, during the processing of these materials, there may be some contamination in the surfaces. So these this contamination should also be controlled when processing these materials, or when processing these devices for the medical application. Also, one important aspect about the surfaces which is different from the bulk is that the surface material is often mobile. That means the movement of the atoms or molecules that are near the surface, they respond to the outside environment, and they can move, and they can orient in different directions, and it is highly significant in case of surfaces, and it gives actually different properties to the materials. If I can give one specific example, for example, in response to aqueous environment, if some surface is in the water, it is in the aqueous environment, the surface may reverse its structure and point the polar hydrophilic groups towards outwards, and hydrophobic groups will go towards inwards of the material. And these hydrophilic groups then interact with the water. So this property, it is shown in the next slide. As you can see in this picture, this polymer poly 2 hydroxy ethyl methacrylate, this polymer, it has some hydroxyl groups and methyl groups. When this polymer is in air, so these hydrophobic methyl groups are outside, and they interact with the air. When this polymer is when it is immersed, or when it's put in the water, upon immersion, the hydrophilic groups, they come outwards, they orient outwards, and hydrophobic methyl groups, they orient in inwards. So there is an interaction with water, because of these hydrophilic hydroxyl or hydroxy groups. And also this change in orientation is also reversible. If we take this polymer, and we take it out from the water and put in air or some hydrophobic system, the hydrophobic groups will come outside and hydrophilic hydroxyl groups will orient towards inside. So in the these atoms in the polymer backbone, they are there in the polymer backbone. They do not move from the place, but they orient itself from outside to inside, and to interact with the different different environmental conditions. When talking about studying the surface properties or studying the surfaces, one important question is how deep is the surface? How deep in the material we can call it the surface. So it has been defined as the surface is a zone where structure and composition is influenced by the interface, and after this interface, if there is a different if there are different composition and there is a different structure, and after that the bulk of the material starts. So the surface has a different composition and structure as compared to the rest bulk of the material. Some of the examples are given. For example, in a polymer, the surface is extended, in some cases is extended to 10 nanometer, and in some cases it is extended to 100 nanometer depending on the polymer. And also in case of gold, an example is given that after 5 atomic layers, that means 0.5 to 1 nanometer, the composition becomes uniform. That means at the surface, at the surface composition is not uniform, it is different. But after 5 atomic layers or after 1 nanometer, the composition of all the bulk is uniform. So we can define from this that if the composition is not uniform, so after this depth it is the surface, and after that if the composition is uniform, so we can say that after this depth it is the bulk. Now the bulk of the material has started. And the interface between this bulk and the surface, it is called interface. To study the surface, there are some of the important parameters which need to be studied, and after studying these parameters we can understand we can understand how the surfaces will react with the biological system, and what should be the biological response to this surface after studying these properties. So different parameters are listed here. For example, surface roughness, how rough is the surface, uh, and surface wettability. It defines as the what is the contact angle, what mm, it explains the how much the surface is wettable and how much surface gets wet after the aqueous phase is put on it. Surface mobility, like I said, this surface of the material is mobile because the atoms and molecules, they move from one position to another position. So these are all self-understood terms. For example, surface mobility, chemical composition, electrical charge, crystallinity, heterogeneity. Heterogeneity means that if there are different types of molecules or atoms that are present in the surface is heterogeneous. So all these parameters should be studied to know to conclude what will be the effect of these surfaces on different physiological systems, and or how different physiological systems will react with these surfaces. Some of these properties that I have mentioned, some of these parameters are also mentioned here and also shown in this figure. For example, in figure A, surface roughness is shown, and the surfaces they can be rough, or they can be stepped, or they can be smooth. In this B part, surface chemistry is shown, that means the surface can be made of different atoms or molecules, or different macromolecules. That it shows the heterogeneity of the surface. If it is made of different types of molecules or atoms, the surface is heterogeneous. In the C part, it shows the homogeneity. For example, these surfaces are inhomogeneous surfaces. They may be structurally, or they may be on the base of composition, they may be inhomogeneous. For example, You can see here, for example, this shaded and this lighter shades, they are not oriented, they are not homogeneous, they are inhomogeneous, they are randomly arranged, these two shades. They may be structure or composition, but they are not homogeneous. So homogeneity of the surface is also important for the interaction with the different biological systems. So this was I was talking about inhomogeneity on the surface, on the surface layer only. On the upper layer there is it is not homogeneous. On some patch there is a one material composition or one structure, and on other in another patch there is one structure or composition. And in D part it is shown that surfaces that are inhomogeneous with depth, that means at one layer the surface is homogeneous, all the atoms are homogeneous, but when we go in the depth, that deeper layers are not homogeneous, does not correlate with the upper layer, but one layer is homogeneous. And in this D part it is also shown that surfaces that are overlaid with the thin film, for example in the surface there is the thin film of another material which has a different properties or different composition or different structure. Surfaces may be also highly crystalline or disordered. For example, this surface is crystalline, there is a crystal structure, or each crystal is made up of some particular orientation or some particular unit cell. But in this lower case, there is a disordered orientation of the atoms in this material. And here in this figure it is shown that even the crystalline surfaces which have the ordered arrangement, they also have different orientation or different ordered structures, or they are in crystalline also. So overall, studying these parameters of the surface are very important, to know their interaction with the physiological system. And the more parameters we study, and more we can determine the properties or interaction of these materials in the physiological system. As I said, that the surfaces are highly contaminated, or they are very prone to contamination because they are close contact with the environment. So for surface analysis it is very critical to prepare the sample. So sample preparation is very important for the surface analysis of the materials. So the materials for the surface analysis, they should be identical in each test, and they should resemble to each other as close as possible. In each test, but each batch of material should resemble each other as closely as possible, and there should be the proper shipping and storage of these materials when they are sent for the tests. The packaging material which is used for packing these materials, these packaging materials should not cause any surface contamination to this material. For example, some of the materials like paper or some polythenes, they transfer the material, and they transfer the material on the surface, but this should not happen because it will change the surface properties, and alter results may be achieved. Fingerprints can also alter the different surface properties. So for sending the samples for surface analysis, fingerprints should not be there. Proper care should be taken so that surface should not have. And the packaging material that are used for packing this, for example, many brands are using aluminum foils for packing different specimens or different materials, but some aluminum foils are treated with citric acid, and this citric acid can contaminate the wrapped biomaterial, and can change the surface properties of the material. Some of the general considerations should also be kept in mind before sending the samples for surface analysis. For example, and in some cases the analysis method alters the surface. So in that case, different analysis methods should be chosen, or use of one or more analysis methods should be done, and the results from different analysis methods are compared. If the results are same, then in 2 to 3 analysis methods, that means the analysis method is not altering the surface. But if the results are not same, results are contradictory from different analysis methods. So in that case, third or fourth method should be used to analyze the results or to analyze any property. And also on the basis of different samples, analysis method should be chosen. For example, as compared to metals, ceramics and glasses, organic and polymeric materials are more easily damaged by surface analysis. So on the basis of the material of choice, on the basis of that, surface analysis method should be chosen. So all these considerations should be taken into account before sending the material for surface analysis. Here in this slide, I have enlisted some of the analysis methods that are used to analyze the surfaces. For example, like contact angle method, which is used to measure the surface wettability. Scanning electron microscopy, or FTIR, scanning tunneling microscopy, atomic force microscopy, electronic ion mass spectroscopy. These all used to analyze the surface properties. I will talk about this in the next slide in detail. So all these methods which are listed here, they are most commonly used methods to characterize biomaterial surfaces. First is the contact angle measurement. Liquid wetting property of the surface. It is used to estimate the energy of the surface, or it is used to estimate if the surface is hydrophilic or it is hydrophobic. Those properties of surface are measured by this contact angle measurement. And depth analysis of 30 to 20 angstrom is the depth analysis of this contact angle measurement. And it has a very low and high dependence on the chemistry. On the basis of chemistry, the surface, this contact angle may change because of the interaction of water molecules with different molecules present on the surface, or different atoms or molecules present on the surface of the material. So if the contact angle is less, if the contact angle is less, the surface is more hydrophilic. If the contact angle is less, that means the droplet of the water, it is more dispersed in the surface. And if the contact angle is more, that means it is not more dispersed on the surface, and that means the surface is more hydrophobic. In the second method, ESCA, it is the electron spectroscopy for chemical analysis. In this case, the electron beam is bombarded on the surface, and in it the X-ray beam is bombarded on the surface. In X-ray beam induces the emission of different electrons with the characteristic energy, and this energy of characteristic energy of these electron beams are measured to analyze the surfaces. And these X-ray, actually it can penetrate the surface up to 10 to 250 angstrom. So the surface up to 10 to 250 angstrom depth can be analyzed from this technique. And in this another technique, Augur electron spectroscopy, in this case the electron beam is stimulated, and it is bombarded onto the surface, and it stimulates the emission of Augur electrons. And the depth analysis of this technique is 50 to 100 angstrom. Another technique is the SIMS, secondary ion mass spectroscopy. In this technique, ions are bombarded and ions are bombarded on the surface, and secondary ions from the surface are emitted out, and the properties of these secondary ions are studied, and on the basis of that, surface properties are studied. In case of FTIR, IR radiations are bombarded onto the surface, and these IR radiations, they are absorbed by surfaces, or they are absorbed by different molecular vibrations which occur in the surface. And on the basis of this molecular, on the basis of the IR radiations that are absorbed, so some radiations are absorbed and some radiations are emitted. On the basis of that, we can say that what type of surface is there. We can know the surface properties of the material. And another one is a scanning electron microscopy. In this technique, and secondary an electron beam is bombarded into the sample, and secondary beam is emission of secondary electron beam occurs, and this electron beam gives the data about the surface of the material. So all these techniques are used in the laboratory settings or in the different industrial settings to study the properties of surface properties of the materials.